today about a hands-on approach I've developed to teaching online and non-majors laboratory for a botany course. I'm going to motivate this by talking about why is online laboratory a good idea. Well, for general context, we've seen year-over-year growth rates of 5 to 15% in online teaching. In contrast, in-person teaching has had stagnant or even declining enrollment. As a result, if we want to reach more students, one way of doing that is using this online context. And we might think of online courses as mostly being at private, for-profit institutions. That's not true. Most of it is actually at public universities or at private nonprofits. So the kinds of places that you and I teach at. And what kinds of students can we reach if we teach in an online setting? Well, we reach a lot of non-traditional students. One provider of course kits for online labs reports that about 70% of their students are women, and most of these women have families and are also working professionally. So these are students we'd otherwise miss. Also, students with disabilities, in some cases, do better in an at-home setting than in a classroom setting, so we can reach that audience. There are increasing online-only programs where the class is going to start, or the entire degree rather, is in an online setting. If those have a laboratory requirement, then somebody has to teach that. I say, why not have that be a botany course? And are online laboratories effective? If they weren't, then I would drop this idea. There's been a fair amount of research, or at least several studies that compare online and face-to-face courses. And what they find is usually that there's no significant difference. If there is, then it's usually a small difference, sometimes in one direction, sometimes the other. So there's not a consistent difference in performance or learning, basically whatever measure is used. And Beal and Brame reviewed these studies recently if you want to see more. Successful online laboratories, sort of best practices, according to people who have done this, they should, just like in person, include inquiry. They should have collaboration between students, as well as interactions between the students and the instructor. That is hard to do. There needs to be self-reflection and self-assessment. There needs to be hands-on, so experiential learning, or remote labs. This would be something where a student is controlling an actual microscope, even if they're not sitting at it, as opposed to simulated laboratories where there's a web page and they sort of pretend to do something. They pretend to pour water into a plant pot. These experiential labs, there's the possibility of doing something wrong. There's more decision making, students have more agency, and they learn more. This is a student whose coleus plants died. I told him, you still have to graft something. He grafted 2 different kinds of brassica together. Seems to work. And why botany? You all don't need much convincing here, but in the BSA Botany for the Next Millennium report, the report concludes there are 2 salient environmental issues of our time: global climate change and the loss of biological diversity. So we say we need to research these, but additionally the report concludes there needs to be more attention to education and communication. So how can we reach more students? Well, we can go where the students are going. At my university, developing this online course allowed me to offer a new course in botany, something that we couldn't do at many universities otherwise. And also the course fills instantly. There are many digital tools that we can use in teaching botany labs. I think there's an entire session on this topic, so I'm just going to list some ideas. Digital herbaria, niche modeling software online, citizen science things like the National Phenology Network, DNA Subway if you get adventurous and want to teach phylogenetics, iNaturalist, so an app that allows you to do both identification and share your results with others and with scientists. A little bit about the course I'm teaching. It's a laboratory course that accompanies an online lecture section 2. Together they're 4 credits, and importantly, they fulfill the gen ed requirement. It's asynchronous. I'm never online with my entire class at the same time. And I've taught it in a 14-week semester. I'm right now halfway through a 4-week semester. And I do a lot of the things you would do in the general survey of botany course. Chemistry, field botany, taking care of plants and growing things, as well as digital botany. And the skills I teach are the same skills I would teach in person. Careful observations, using dichotomous keys, creating and testing hypotheses, interpreting graphs, communicating results, and following directions, as well as taking responsibility for a project. I'm going to give you some examples of the lab activities I use. I can't hit them all, but I'll do about half. So we do DNA extractions, and we used to just extract, but now I've made it hypothesis testing by having students compare the amount of DNA extracted from rice germ and wheat germ. I motivate this by talking about the size of the rice and wheat genomes and diploidy versus hexaploidy. Chemistry of life, they do simple pigment extractions using different solvents, water, alcohol, and mineral oil, and they do it from different colored leaves and petals, and they can make hypotheses about what kind of pigments are in which, based on which solvents work and which solvents don't. Field botany, we do this in a few different ways. We do gymnosperm identification. This is largely based on photos I provide, but also based on plants they find. They identify mosses, liverworts, and ferns, and they do this basically using iNaturalist. They do winter twig ID and summer tree ID. I'll talk about those. For summer tree identification, they have a dichotomous key for the region, and they identify several trees. They then upload pictures to iNaturalist, and they compare the results. If there's a discrepancy, I send them to a website to settle the disputes. They then give both the key and the app a score. Turns out the app works better because the key doesn't have some recent invasives. For field botany, in the winter I collect twigs. These twigs are part of their lab kits. And they then do a couple of things. They use a dichotomous key to identify them. I embed the key into a course management software quiz, such that if they get the wrong answer for a step, the next question redirects them to where they should be, so they can eventually get to the right answer even if they make mistakes along the way. I do that in class too, in person, and it works well. They also take photographs of parts of one particular twig. They describe its traits, everything, bud scale, scars, number of bud scales, things like that. For plant care, they make terrariums with 2 different bryophytes, a moss, a liverwort, as well as a spike moss. They grow them in a bright environment and a shady environment and compare the results. They grow coleus throughout the semester. They make cuttings and they graft coleus together. That's my graft right there. They also grow fern gametophytes in the petri dish, just like I would do in person. And eventually they add water, let fertilization happen, and grow sporophytes. We talked a bunch about climate-related issues. The first thing they do is identify trees in their yard, they map them, and they then use an online app to calculate the energy savings due to reduced cooling costs of those trees, and they calculate carbon storage by those trees. We use the phenology network, citizen science, to look at things like how does leaf emergence time change with latitude. And they get to do some troubleshooting. What about these points up here? Do we trust them or not? They do biogeography. So they model plant distributions. And I think I'll talk about that in just a second. Genetics, this is the one thing where I use a traditional kit provided by a commercial supplier. And they look at 3 different generations of radish, looking at inheritance of anthocyanin and dominance. For supplies, this is the hardest part of teaching this course. I gather all of the supplies, box them up, and mail them. It costs about $60 per student Things like the hand lens and timer are a few dollars each. Many of the other things are pretty cheap, but require some time to collect. They get this entire collection. It's hard to mail living plants. It's about a 60% success rate at this. So if they live within driving distance, they're required to come pick up. I use a program called Padlet that I and they love for submitting assignments. Each student gets their own column for each assignment, and all of their descriptions and photographs go in that column. This is something that allows self-assessment. For example, we can see that this student did pretty well, but when this student submits her pictures, she can see that maybe she's not doing the same thing as the student next to her. And I also just give direct feedback here about how they're doing. They can see that, and their peers can also see that feedback. Challenges of teaching in this way, they're not small. Making those lab supply packs is time consuming. Shipping live plants is challenging. We talked about that. It's really important to respond to questions almost immediately, because if a student is in the middle of a 2-hour exercise and it's 10 at night and they have work the next day, then I can't really say, wait for me to email you tomorrow and then you can start again. So I have given them my text number, my phone number, and my email. I use a Google phone account so I can just cancel it at the end of the semester or if there's a problem. But for the most part, they will just email me. Online in this case is not an excuse for large class size. In fact, my university generously caps this course at 16, smaller than we would cap an in-person laboratory, and that makes it a little bit more manageable. Future directions. I would love to think about research collaborations because I would like to test whether this online course leads to high levels of student engagement, learning, and positive attitudes, but I don't teach it in person. So if anyone who does teach a similar course in person wants to collaborate, we can talk about what that would look like. Also, dissemination. I have spent a lot of time developing this material, and I would love to see it used. I haven't figured out a great way of doing this yet, or haven't found the time to do it. But if anyone is interested, I am happy to share what I have, and I can start with just emailing stuff for now. So please let me know. I want to acknowledge my university, the University of South Carolina Upstate, for providing funding that allowed course development and allowing me to teach this course. I think I might have time for questions.
As far as the field part, do you get students outside the geographic range of South Carolina?
Oh, so do not let students outside the country enroll in the course because you can't ship the supplies.
Okay.
Live twigs, big problem. So I do, if I had a student in Ohio, it worked fine. I just found a new key for him to use. The student in Germany, we had to design all sorts of accommodations for him. Maybe one more?
Maybe one more, yes.
Have you had any experience with students trying to cheat? I gotta ask.
The assignments are all designed in a way that cheating doesn't mean anything. Like, if they're doing a flower dissection, oh, I didn't talk about the stickers. Do you see the sticker in this photograph and the sticker in this photograph? Each student has a unique sticker, either a different color or different shape, and so I know all of the pictures are theirs. And so if they've done the dissection, I can tell it is their own.
That's brilliant.
Nice.
Wonderful. Thank you, Ben.
Everybody writing
Let's
down
give a
stickers.
round of applause.